Okay, great. Okay. Welcome everyone. So today we'll talk about the advanced topics of LabArchive, particularly LabArchives Research Edition. We'll start with a slide deck and then we'll transition to the live demo and talk about templates, widgets, and some of our integration partners. So this slide shows our products we have in LabArchives in terms of which product Medical College of Wisconsin has as their enterprise license. That will be Electronic Lab Notebook, which is used to manage your research notes, your data, and as I mentioned, will be the main focus of our advanced topic today. So LabArchive supports of the pillars of a well-formed data management plan, which is often a requirement for grant-funded research, particularly for NIH grants. When we talk about templates, you can establish a data management plan through our templates, and templates will help you organize your page and fit your policy and process and workflow. And also what's great about templates is that you spend less time putting important information, like maybe your purpose and objective and procedure and so forth. So you have more time organizing and, excuse me, you have less time organizing and you have more time working on your experiment design, which is more important. And also what's great about templates is you're consistent among your team. So if everyone will use the same template, and if you need to, let's say, see data from months or years ago, you're able to find it. You're able to ensure your entries are complete because with your heading, you can tell what should be um, what should be highlighted under that which text entry or what is something under the attachment. And as I mentioned, you save time when you establish a template. Also, we see a lot of teammates establish a template notebook. So how you can do that is when you create a new notebook, label the notebook as template notebook, and then under the notebook manager, which is found when you click on the notebooks link and click on the notebook stack. In the notebook manager, you have an option then clone that template notebook for maybe your project or maybe your team member, establishing everyone on the team to be consistent and also assure time. So as I mentioned earlier today's session, we'll talk about templates and widgets. So this slide shows the difference between the two. So in templates, you have a standard pre-formatted entries, including attachments, rich text entries, and headings. And then with templates, you can easily make changes if necessary. But with widgets, these are custom HTML forms with interactive elements such as checkboxes, radio buttons, drop-down menus, even calculated fields, for example. And to create a custom widget, you have to go to the widget manager, which I'll demo you doing a live demo. And unlike templates, widgets requires more time to create and update. So the widget manager, this is how you create your custom widget. The only people or only roles have access to the widget manager are the notebook owner and the notebook administrator. When they establish the custom widget, then everyone on the team, including the user and guests, will be able to have access and use the widget. And finally, before we transition to live demo, I want to take time to show you our great support options you have, which is especially important, particularly if you want to establish your custom widget. So we have a wonderful global support team ready to assist you with any questions you may have, like for example, when you create your custom widget, or even if you need to maybe have additional training on custom widget as well, they're happy to help you. We have teammates based in the US and the UK and Australia with close to 24-hour support desk coverage. We also have a help center where you can find great articles about widgets and also how to establish templates. And finally, when you go to LabArchives-resources, this is where you can find links to tutorials, quick start guides, and our webinars. With that being said, let's now transition to the live demo. But before I do that, are there any questions? Okay, so now we're in my notebook. First, I want to talk about templates and show you some example templates you can use for your notebooks. So I created a templates folder where I created different templates. So first off, I have the traditional lab notebook where you can see this as, I guess, a format for paper notebooks where researchers are supposed to format in their paper notebook, where you have your purpose, your protocol, where you list your experimental entries or data, your results that you see in your data and your references. And also with the template, you're able to attachment, your file, such as maybe in this case, your protocol. It could be maybe a picture of the equipment that you're using in that experiment and so forth. With templates, I consider them to be discipline agnostic in terms of you can have any discipline and create a template out of it. So we have, for example, a template used for clinical research, for example, where they create under headings. This is where a researcher would put the location of primary source or raw data, where is it located in their local drive, what software they used, additional notes and data dictionary, which I find very important because this is where, let's say they add abbreviation, important abbreviations, and they don't remember where it is, they can able to use the search um, the search feature, type in maybe important keywords, and they're able to find that abbreviation. Output, data set storage. If it's stored outside LabArchives, where it is. And then finally, you're able to attach your code and scripts either in a text file, or we have in a entry type called plain text where you can paste in your R code script, for example. And finally, output files. And then lastly, you can also create a template for your protocols that you use in your experiment. So for example, I created a genotyping template where I added my mice ID, date of birth, purpose, objective, protocol, my results. I also have immunohistochemistry template where I have my reagent list, my protocol, where every time I use this template, I can then type in the X of how many times I use this particular step. And also I can also create a custom widget as well, allowing me when I use this custom widget, check mark each step I completed. And finally, I create a section for my images where after I finish the protocol, I know where I can put my images. So to use a template is very simple. So I'm going to, under my microglial and depression folder, under September folder, I'm going to select new, and click Copy Existing Page. If I, for example, have a template notebook of another notebook, I can click Copy from another notebook to retrieve the template and use that template. So for this case, I'm going to select Copy Existing Page. And since this is my genotyping folder, I'm going to use my genotyping template. I can then select which entries I can include. So I'm going to select all, then click Continue. And then I can then enter the name of my new page. So in this case, I'm going to type 31 GY and then maybe my mice ID. So I'm going to say once I click create a page, notice that the template I use is copy over, and all I have to do is type in my information. And so forth, and then add my results as well. So speaking of results, I'm going to transition and talk about our integration partners. But before I do that, are there any questions about templates? Okay. So we have our integration partners. I'll show you how they look like on a page. So for example, we have SnapGene and GraphPad Prism. So let's talk about SnapGene. So with our SnapGene integration, after you attach your files to your notebook, we have something called Docker Viewer, which allows you to view your SnapGenes while you're in your notebook. So here I can see my DNA, my map, my sequences. Apologies, my computer is kind of slow. My enzymes, my feature, and my primers that I use for this particular DNA. On the left-hand side screen, this is where I can see my enzymes, my features, my primers, my translations, and so forth. This is good because now, let's say my work laptop only has the SnapGene license, but today I'm working at home, so my personal laptop does not have the SnapGene license. With the SnapGene Docs Viewer, I have the opportunity to see my SnapGene files and also my colleagues' files. And if I have a question of for my colleague about their SnapGene file, I can use the comment feature and direct them a question or a comment. Also, with our SnapGene integration, you have an opportunity to send over your SnapGene files from your SnapGene desktop to your notebook. So I have my SnapGene desktop open. To use integration, you go to SnapGene for Mac, and I believe for Windows, you go to File, and then click Preference. And under Preference, you go to Files. And then you'll see a place where it says enable file exchange with an electronic lab notebook. And here, this is where you can type in your username, so LabArchives email address, and then your password. Because Medical College Wisconsin uses SSO integration with LabArchives, you need a password token. To retrieve a password token, go to your name, click External App Authentication, and then click Copy. And then you can use a password token for not only your Snapgene integration, but also your GrabPad, Prism integration, and Microsoft Office plugin as well. Okay, so let's exit that out. So now that I signed into my LabArchives account, when I go to Files, I have opportunity to open files from LabArchives. So let's use the Snapgene file that I showed you. Give me 1 second. I have a lot of folders. Okay, so that was the file that we saw. So I click open. You'll see my file, and I can make changes to this particular file. So I'm going to change this color, maybe to red. And now I can save this file to both my laptop and also to my notebook. Okay, when I click save, click replace, and you see the file is saved successfully to my notebook. And when I click OK, before I go back to my notebook, I want to show you how you can save new files from your Snapgene desktop and also save it to your notebook as well. So I'm going to use this one, for example, and I'm going to click Save to LabArchives. I can then, if I need to, change the name as well. So I'm going to add LabArchives. I then decide what page I want this file to be in. So I'm going to put it under GY September 30, 2022 page. And when I click save, and it saves successfully to my notebook. And now when I go back, if I scroll down, you'll see the Snapgene that we updated with, I think it was orange prior, now is red. And also we see the sequence. And when I go to its vision history, you'll see all the changes that I conducted in the Snapgene file, including how I changed the color. And if I go to revert to its version and click OK, not only the orange color returned, but then if I go to its vision history, you'll see that action has been archived. So then now let's talk about GrabPad Prism integration. So similar function in terms of you're able to open files from LabArchives to your GrabPad Prism, and also you can save your Prism files from your Prism desktop to your notebook. And same thing as well, you use your LabArchives email address and then use the password token, which you're able to retrieve when you go to your user icon and then click external app authentication. Also, when you click on LabArchives, you're able to see all the GradPad Prism that are in your notebooks. So you also see recently saved as well. So let's say I want to use the step, if I want to open demo graph, click OK. And I'm going to add more data to this. And now when I click under, so this in the GradPad Prism, there's a column called LA, and this is how you can use integration. And because this file is already in my notebook, I'm able to decide if I want to archive it to the inbox as a new file or archive it as an updated file. If I click this option, I have the opportunity to write what change and also have a description field as well that I can type into. So I'm going to choose archive as an updated file, and then I'll type I added more points. Okay, and same thing, I'm also going to send in a new Prism file to my notebook as well. Because of time, I'm going to use the example data set. Okay, and after I save this Prism file to my laptop, oh, let me hold a second. Okay, perfect. And then go to the LA column and click LabArchive it. I can, as I mentioned earlier, I can write in a subscription if necessary. And when I click archive and go back to my notebook, first I want to show you the updated Prism file that we revised. And same thing applies if I go to Vision History. You'll see all the changes changes that have been done to a certain Prism file. Notice how under change, you'll see the comment that I wrote, I added more points, all appeared in my notebook. And when I click on the timestamp, you'll see the difference. And if I click to revert to the version, it'll go back to the prior version, and that change will be documented in the revision history. Keep in mind, though, if you revert to earlier versions or make changes to this particular file, same thing with Snapgene, it will reflect in your notebook, but will not reflect in your Snapgene or your Prism file in your local laptop. But if you make changes in those files in your Snapgene desktop and GradPad Prism desktop, then those changes will be reflected in your notebook. So pretty much is a one-way One-way integration, not a two-way integration. And then every time you send over to send over Prism files, it'll go to here. This is your LabArchives inbox. And LabArchives inbox, this can be used to send files to your account via email. And also, as I mentioned, this is where you'll see your third-party application files, such as your GrabPad Prism. And also in LabArchives inbox, you can establish inbox rules as well. So here I have my GrabPad file, and in the triple dot menu, this is where I can move this particular file to a particular page. So I'm going to move it to this page. And when I scroll down, you'll see this GrabPad file was successfully moved to this page. So that was our main integrations we have. If you want to learn more about our integration partners, when you go to the info icon and click knowledge base, and you scroll down, are you able to see all our integration partners that we have, including if you need a reminder how you integrate with Prism, Snapgene file is there. And also we integrate with Jupyter, Protocol IO, RedCap, for example, as well. So I'm going to put this into the chat. If you want to have it as a reference. Okay, so now we talked about templates, we talked about integration partners. Let's now talk about widgets. Are there any questions? Okay. So as a reminder, to use a widget, you go to the +2 menu, and when you click widget, this is where you see all the widgets you have, including the built-in widget and any customized widget you have access to. So I have on this page example widgets that are embedded in this page. So I have a chemical sketcher widget, a concentration calculator widget, PCR calculator, a freezer box widget that you're welcome to use as a well plate. And also I have a custom widget where a hospital created to establish a consistent workflow whenever they conduct their cell counts. So to create a custom widget, you'll go to the widget manager. As I mentioned, only notebook owners and notebook administrators have access to. And under the widget manager, you have all your available widgets. The built-in widget will have the LabArchives logo, and the custom widget will have the heads and shoulder logo. When you create a custom widget, click New. This will copy a custom widget or built-in widget. This is especially important if you are maybe updating a custom widget. A good idea is to copy the widget you're working on, and while you're working on that custom widget, your colleagues will still continue using the other widget. And delete this as it says, it just deletes the widget. Um, keep in mind when you delete a widget, it'll be deleted permanently. You cannot bring it back. So I'm going to make a widget. So I'm going to title this October 31st. And I'm going to make an electrocardiogram widget. So I'm going to abbreviate ECG widget. In the widget manager, you have 3 tabs. The widget HTML editor. This works a lot like the widget text editor, which allows you to format your widget and add interactive elements. The preview tab, this allows you to view and test your widget before adding it to your notebook, which we recommend you do. And the script editor, this allows you to view and edit the script that makes up your widget. And if you're experience in JavaScript, to create your JavaScript widget, you would use a script editor. So for today's session, we're going to use the HTML editor tab. So I'm going to type in ECG results, and like a which text entry, I can change the font, maybe the size, add color, for example. Oh, maybe I should not highlight that. So my ECG widget, I'm going to type in object animal dash animal ID. And next, I want to add a text field. So my colleagues and myself can type in the subject and dash animal ID. So then I would go to this text field. Any field that you use under the name, you need a unique lowercase name without a special character. So in this case, I'm going to type in subject. Then we have the value field. This can be used to find a default value, like a number, for example, or customize the field comment. You'll see more about field comments momentarily. So when I click OK, now I have a field for people to type in subject or animal ID. For this widget, I want to also have forms completed. And then I can use checkboxes, for example, which will be in a checkbox. And then as I mentioned, your unique name will have lowercase name without special characters. So in this case, I want one, maybe one for medical. So then I write medical history. Maybe want a checklist for my colleagues and myself to select when we complete the eligibility form. So I'm going to type in and then type in eligibility checklists. Maybe want a checklist, a checkbox um, for my colleagues and myself to select when we complete the monitoring log. And then for this widget, I'm also going to create a section for my results. Beats, time, and beats per minute. Okay. Okay. So then I'm going to establish form command. So under beats, I'm going to type in beats underscore number. So every time you want to um, establish a command, you have to write in your unique name and underscore the command. So the number, this means that every time someone types in something here, it has to be a number. And then under value, I'm going to type in 60. So this means that every time someone uses this widget, the number 60 will appear. And then finally, for my file form command, I want to use a calculator to calculate beats per minute. So they use beats underscore calculator. So to perform calculation using form command, you need hard to type in your calculation before copying and pasting it to your to the value. So you need a pound key, excuse me, a number, symbol, and then squiggly line, and then type in the unique names that you want to use for calculation. So for this case, I want to use my beats and time. So then I would use beats number and then have another squiggly line. And then I want to divide that by parentheses. Then I divide by that time underscore number, and then I divide that by 60. And once I'm happy with my calculation, I then copy my formula and paste it under my value, make sure there's no space. Okay, and then I'm going to type in the unique name. Oh, cannot spell. Okay. So beats, I would beats a formula and click OK. You'll see my formula appears, and then when I click preview, make sure everything is okay, which it is. I then save my widget. Make sure to save your widget periodically, because unlike the widget text entry, the widget manager does not have the autosave. So then I can then able to use my widget. So let's put it in a new page. When I click widget, and if I scroll down, you'll see my widgets there. And then I can type in maybe my subject ID, select the form is completed, and then type in the beats. Similar to the integration, we also have in our knowledge base all the articles you can find about widgets, including what are they, the widget manager, how you can customize your widget, and also feel free to contact our support team at support@labarchives.com for questions while you're creating custom widget, and also to help you brainstorm how you want your custom widget to structure, to be formed. So with that being said, we have plenty of time for questions. Are there any questions about what we talked about today? We talked about a lot. We talked about the templates, we talked about widget manager, how you create a custom widget, and also we talked about our integration partners, which are Snapgene and GrabPad Prism. Okay. I don't see questions coming in, or I don't see questions in the chat. So we can end our session today. Thank you so much for coming. And as I mentioned, if you have any questions, please contact our support team at support@labarchives.com. Thank you so much and happy Halloween.
Thanks, Gloria.
Yep. Thank you.
Bye.
Bye.